Hello everyone. In today's video, we are going to discuss the principles of inheritance and variation revision. Okay? Now, in this chapter, we have few terminologies. First, inheritance and then variation. So what do you mean by inheritance here? Let us consider one character. Say we have the eye color. Now, from parents, when this eye color is passing on to the progeny. Progeny is nothing but the children. Okay. So from parents to progeny, whenever any character, say eye color, is being passed on, this process of transferring of character from parent to progeny is called inheritance. Yeah. So process by which characters are passed from parent to progeny is called inheritance. Next, we have variation. Now, if you observe the progeny of any species, let us say humans. Now, this progeny is not 100% similar to their parents, isn't it? We have some dissimilarities. The features will be different. So that differences what you see in the progeny compared to your parents is called variation. So variation is nothing but degree by which progeny, that is children, differs from that of their parents is called variation. Clear? Next, we'll talk about the scientist Gregor John Mendel. Gregor John Mendel was an Austrian monk. Please remember, it is not Australia, it is Austria. Clear? And he has done experiments on Pisum sativum, which is otherwise called as garden pea. Please don't get confused between garden pea and sweet pea. Sweet pea was not taken by Mendel, only garden pea, and the scientific name will be Pisum sativum. Okay. Year is again important given in your textbook. So 1856 to 1863, we can say 7 years of experiment he has done. Now, what were the reasons for Mendel's success? These are the few points we have to remember. He has applied the statistics and mathematics in his experiments. See, whenever we use mathematics, we'll get some numbers, isn't it? So numbers will be more valid than, vague answers. So he has used that numbers and statistics in his experiment. Then he has used a large sampling size. What do you mean by sampling size? In order to do his experiment, he didn't do only in 50 or 100 pea plants. He has done that experiment in thousands and thousands of pea plants. So if you're doing experiments on thousands of organisms, now we can make that, whatever results we get, we can generalize it. If you do experiment only on 10 or 15 individuals, we cannot generalize our results. So because of having, since he has done in thousands of pea plants, we can say whatever results Mendel has got, we can generalize those results. So the large sampling size was one of the advantage in the Mendel's experiments. Next, coming to the year of publication, 1965, again important. Now, why Pisum sativum? See, Pisum sativum made Mendel lucky. Okay, so what were the advantages of selecting Pisum sativum that we are going to learn here? There are 4 advantages. First, it has expressed, whenever you consider the Pisum sativum, it will be expressing the contrasting traits. What do you mean by contrasting traits? If you consider any character, for example, if you take height of the pea plant, okay? In this, we can have either tallness or dwarfness. Clear? The pea plant will be either tall or dwarf. We don't have the in-between heights like how we have. Humans have a range of heights. We have very tall, very dwarf, and in-between average and all those things. But pea plant will have either tall plant or dwarf plant. Clear? So that is called contrasting. Contrasting means opposite. So because of that, this was one of the reason. Next, the pea plant have short lifespan. Mendel need not have to wait for many years. Say if the pea plant lifespan was say 12 years or 30 years, okay, by the time it is producing the progeny, that is children or seeds, Mendel's half of the life will be over, isn't it? So the pea plant has a short lifespan is one of the advantage. Next, it has got bisexual flowers, which can help him for both cross-pollination as well as self-pollination. So he can play with that. And also, whenever he was getting progeny, he was getting large number of seed at one time. Clear? So these were the four reasons of selecting pea plant in his experiment. Next, we'll discuss what were the reasons for his unrecognition during Mendel's time. See, during Mendel's time, there was, we didn't have the communication, like we didn't have the cell phones, we didn't have the social media, like what we have nowadays. So because of that, that is communication problem, he could not, publish his research works all over the world. And he could not explain what exactly the factors are. Whenever he was giving the laws, he was explaining everything in terms of factors. But when someone was asking, Bhaiya, what is factor? So he was not able to explain exactly what the factors are, where are they present, okay, how they are actually getting transferred from parents to progeny. He was not able to explain all those things. Next, concept of factors not blending was not accepted. Now, what do you mean by this? See, Mendel has given law of dominance, isn't it? According to law of dominance, what is it says? If the parent is tall, or dwarf, and dwarf. Okay, in pea plant, parent is tall and dwarf. According to law of dominance, the progeny will be tall. Okay, we don't have the dwarf in the progeny. So out of these two characters, only one character will be seen in the progeny. But in those days also, we have other plants. Okay, say for example, a plant with red flower and a plant with white flower. Both of them belong to same species. So red flower and white flower. Now, when both of them were crossed, the people were getting pink flowers. Now, how is this possible? Now, pink, if you are getting, that means we see the blending of red and white there. Okay. So, whenever the Mendel was explaining the law of dominance, other people were, telling, then what about the blending concept? How come we are getting intermediate character in the progeny? So, this also Mendel could not explain there. And also use of mathematics in biology. See, we biologists, usually we don't like maths. That's why we are in this stream, isn't it? So there also, since Mendel was using maths in biology, many people were like, no, we cannot take that. Okay. So these were the reasons for his unrecognition during his time. And again, like after Mendel's death in 1900, three scientists have come up with the results, whatever Mendel has got. And they came to know that whatever results they have got, Mendel has already done those things. Okay. So you have to remember who are those three scientists: Correns, De Vries, and Sharma. So Carl Correns, De Vries, and von Sharma in the year 1900. This you have to remember. Clear? Next, we'll study the 7 contrasting traits in pea plant the Mendel has selected. So here this table is very, very important. Mendel has taken 4 things. We can say 4 things: plant height, flower, fruit, and seed. Like this, if you remember, it will be very easy for you to remember things. Okay. So for plant height, we have only one character, that is height of the plant. So tall and dwarf. Tall will be the dominant. I'll tell you a trick to remember this. Yeah, later. Now, if you consider flower. So in flower, we have position of the flower and color of the flower. Position, dominant is axial and terminal. What do you mean by axial and terminal? Say, for example, this is the plant we have with the nodes. Say these are the nodes. Now, if the flower is arising at the nodes like this, then that will be your axial position. Okay. Instead, if the flower is present at the top, something like this, so this will be your terminal. So this is the terminal position, and this is your axial position. Okay. So axial will be dominant, terminal will be the recessive character. Color, violet. We obviously like, bright colors. So violet will be dominant, white will be recessive. Next, coming to the fruit. Shape and color of the fruit, you have to remember. Full. How is the pea plant? Pea, something like this. Matar, pea, you have seen, isn't it? So how is the pea pod? See, fruit, we call it as pod here. So don't get confused. Pod or fruit of the pea is both are same. So pod shape or pod color also we can say. Shape, it is full, will be dominant. Constricted. Constricted will be something like this. So this will be constricted and full. And if this is green, that will be dominant. Yellow will be your recessive. Next, coming to the seed. Now, inside we have seeds, isn't it? Now, seeds, again, we can have the shape and color. He has taken shape and color. Round will be dominant, wrinkle will be recessive, and yellow seeds will be dominant and green will be recessive. For this, I'll tell you a trick. Let us say we are growing a pea plant. Okay. Now, the dominant characters are what we actually want in our field. Clear? So what are those? See, whenever you are growing a plant, obviously we want our plants to be very tall. Okay. So tall plants, let me draw with some other color so that it will be interesting. Okay. Okay, so whenever we are growing plants, we want our plants to be tall. So tall plant will be dominant. Now in this tall plants, when we are seeing the flowers, okay, first you have to remember flower, fruit and seed sequence. First comes flower, from flower we get fruit, and in the fruit we have the seeds. Clear? So flowers. Now since the plant is very tall, we cannot pluck the flowers which are at the terminals, isn't it? So we want flowers at the axial. So axial will be dominant. Clear? Now, obviously, we want bright color flowers. So violet color flowers will be our dominant one. Are you following? So tall plants, then axial flowers and violet flowers. These will be our dominant flowers, dominant characters. Then this flower is getting converted into fruit. Now, how will you like that fruit of the pea should be? It should be full, isn't it? Something like this, not like this. So we want full pod or full fruit. And how should be the color? Green, obviously. When we go to the market, which color of pod you select? Green ones or yellow ones? Green ones, isn't it? So green color pod, we have the dominant character. So full and green one. Now, when you open the pod, all right, when you open the pod, how should be the seed shape? It should be round, obviously. Whenever we think of pea plant, pea seeds, round. So round will be dominant. But at the end, dominant character, you have to remember that yellow seeds will be your dominant. So inside this green color pea pod, let me draw this for you so that you can remember the things nicely. So in this green color pea pod, inside when you open the pea pod, you should get yellow round seeds. That will be your dominant characters. Clear? So this is a very small trick to remember. Funny trick. Okay. And again, this is very important. That's why I'm telling you the trick here. Next, we'll talk about the inheritance of one gene done by Mendel. So here again, I have drawn this in the form of a table. Now what Mendel has done, parents, he has select. Now here the character what we are going to take is height. Okay, I'm taking the height of the pea plant. And whenever he has taken the parents, always he had made sure that he has taken pure lines. Now what do you mean by pure lines? Pure lines are the homozygous condition. Homozygous condition. Now what do you mean by homozygous condition or the pure lines again? See, we have, now I'll go in, a step forward so that you can understand something. See, we have, in humans, if I consider, we have 46 chromosomes, isn't it? We have 46 chromosomes and we say these 46 chromosomes are in 23 pairs. So we have 2 pairs of chromosomes, 23 each. Clear? Now let me draw one pair of chromosomes. Say this is your one pair, that is chromosome number 1. Likewise, I can have chromosome number 2, then chromosome number 3. Like that, I will have 23 pairs of chromosome. Clear? Now, these are the chromosomes. Where exactly are the genes present? We know genes are present in the DNA, isn't it? Now, DNA is forming our chromosome. Clear? Now, let us say this is the position of the genes for height. So, in both these chromosomes, now what is this chromosome called? For your understanding, I'll tell you here. This is called homologous chromosome. Okay, this is homologous chromosome. What is homologous chromosome? Out of 46 chromosomes, as we have said, we have 23 pairs. See, suppose this is a zygote. Okay, what do you mean by zygote? Zygote is formed by the fusion of sperm and egg. isn't it? Sperm and egg will fuse to form the zygote. Now, sperm will have 23 chromosomes, egg will have 23 chromosomes, isn't it? Both of them will fuse to form 46 chromosomes. So here, the two sets of chromosome what we have are from the parents. That is, one from father and the other set is from mother. Okay. Let us say this 23 chromosomes of father, this is the first chromosome, and here also this is the first chromosome in the mother. Now this first chromosome of father and first chromosome of mother, we call them as homologous chromosomes. That is chromosome number 1 of father and chromosome number 1 of mother. Both of them will be considered as homologous. Similarly, second chromosome of father, let me write down with some other color, second chromosome of father and second chromosome of mother, these two will be considered as, again, the homologous chromosomes. Clear? Now, if you go through the terminology here, what do you mean by homologous? Homo means what? Same. Logous is what? Structure. Okay, same structure. So basically, homologous chromosomes will have same structure. Now, what do you mean by same structure here? See, everything will be same. Okay, the set of genes. For example, if I consider this chromosome number 1 of the zygote, that is progeny, in this area, I will have genes, let us say for eye color. Okay. Now, in the same chromosome, I can have another set of genes, let us say for height. Okay. So, here also eye color, here is height for maternal. Maternal is mothers, isn't it? So, let us say, okay, first let me write down paternal and then maternal. So, in father's chromosome, that is paternal chromosome, here I have eye color, gene for eye color. In mother's chromosome also, I will have the gene for eye color. Same way, in the first chromosome of father's chromosome, here I'll have the height, gene for height. Here also I have the gene for height. Are you able to follow? Okay. So, basically, what do you mean by homologous? In both the chromosomes, we will have the same set of genes. Okay, whatever set of genes we have in one chromosome of a homologous chromosome, the same set of genes we will have in the another chromosome also. In this chromosome number 1, we have 2 chromosomes, isn't it? So whatever chromosome, whatever genes we have in this chromosome, the same set of genes will be present in the other chromosome of the parent. Clear? All right. Now, coming to this parents again. So, this inheritance of one gene. Now, why I have written capital T 2 times here? Parents are diploid. Okay, so diploid will have homologous chromosomes. So, in one set of chromosome, let me write like this. So, we have one pair of chromosomes like this. Okay, let us consider this is chromosome number 2. So, in chromosome number 2, of father, okay, or male, let us consider this, okay. So in parents, in chromosome number 2 of father, we have a gene for height for pea plant. And this gene for height, let me draw with some contrasting color, okay. So gene for height for pea plant is here. And both of them are got the dominant alleles. Now, what is this dominant and recessive? I'll tell you later. Here, if I tell everything here, you'll get confused. All right. So here, what is capital T means? In parents, let us consider this is father. This is male plant. Okay. So in male plant or father, we have homologous chromosome. Let us consider. This is just an example. Okay. So in chromosome number 2, we have homologous chromosomes here. In both the chromosomes, we have the dominant allele, that is capital T and capital T. Whereas in mother's chromosome, if you consider this mother's chromosome, okay, in mother's chromosome also the position of the gene will be same. Clear? So where is that position? Here. Both the alleles, both of the genes will be smaller, that is, they are recessive alleles. Am I clear or am I, confusing you? Let me come again. In inheritance of one gene is, I'm taking one character. In this example, I have taken the character that is height of the pea plant. Clear? So parents, I'm taking capital T cross with small t, small t. So I'm trying to explain what exactly is this capital T, where is it present, all those things. Clear? So capital T are nothing but they are actually alleles. Clear? So I wanted to tell you about alleles. Now, what do you mean by allele? Allele is nothing but a type of gene which are present on homologous chromosomes. Okay. Now, I hope you'll understand. What is homologous chromosome in a person? Okay, in a person is a diploid. So here I have shown in humans, we have 46 chromosomes. 46 chromosomes. How do we get this 46? 23 from father, 23 from mother. Okay, both of them put together, we get 46. So in pea plant also, it is a diploid plant. Clear? So there also we'll have some diploid number. There also will have homologous chromosomes. Say, for example, in chromosome number 2, we have the capital T and capital T. Both of the homologous chromosomes have got the dominant genes. So both of the genes which are present in homologous chromosomes, we call it as allele. So this one T is allele, the other T is also allele. So both the alleles will be same. Now, in a homologous chromosome, if both the alleles are same, okay, then we call them as homozygous condition. Clear? Homozygous condition. Why do we call homozygous? That also I'll tell you later. Okay, give you some time. Now, pure lines. How do you get pure lines? See, we get pure lines when you repeatedly self-cross any pea plant, any plants for several generations. For example, if you are selfing a plant for 5 to 6 generations, then the 6th or 7th generation will definitely have the homozygous condition. Okay, what do you mean by homozygous again? Both the alleles will be same. It can be capital T or small t, small t. Clear? So if both the alleles are same, such condition we call it as homozygous. Now, Mendel, whenever he has started any experiment, he had made sure that he will get pure lines only. Okay. Now, what do you get? What do you mean by pure lines? Nothing but homozygous condition. How do you get pure lines? It is by self-crossing, self-pollination or self-crossing between the plants for several generations. It could be 4 to 5 generations. 5th or 7th generation plant will be definitely your homozygous condition. So those are called as pure lines. Understood? So this is about parents. Now from parents we get gametes. Isn't it? From father we get sperms, from mother we get eggs. So in this case also from parents, capital T, now they will segregate and both of them, since both of them are homozygous, okay, so the gametes what we get, both of them will be capital T only. So that is the meaning of homozygous condition. Okay, the pure lines or the homozygous alleles will produce same types of gametes. That is the meaning of homozygous condition. So here, let me erase all these things so that I can explain again. Okay. Now, again, so parents are capital T, small t, okay? And the gametes what we get will be same. So both times we get the capital T here and both the times here we get the small t. Clear? Now in F1 generation, what happens? We have to cross it. So all the generations, all the gametes you have to cross and we get all the heterozygous condition. Clear? So capital T cross with small t, we get heterozygous condition without any fail. This is what your law of dominance say. Okay. Now what he has done selfing of this heterozygous condition. So he has taken heterozygous condition again and he has crossed it again. Now what are the gametes we get? Capital T, from here small t, capital T and small t. Okay, you have to make a Punnett square. Now, this is called your Punnett square, which was given by Reginald C. Punnett, who was a British geneticist. Here, in this Punnett square, when put the gametes on the sideways like this, and you'll get the box there. Now, in this box, what are the things we are getting? We'll get two things. One is called phenotypic ratio, and the other is genotypic ratio. What do you mean by phenotypic? The outer appearance of the plants. So this is the results we are getting. Okay, when you make a Punnett square, these are the 4 progeny we are getting. And if you put, if you look at the phenotypic and the genotypic ratio, phenotypic is the morphology, outer appearance, what we can see. So here you have to follow the law of dominance. That means whenever we have capital T, there we can have the tall plants only. So here we have tall plants 3 times and only once we have the dwarf. Clear? So tall is to dwarf will be 3:1 because tall will be like this. Clear? And dwarf is only 1, small t, small t. Next, if you talk about the genotype. So genotype, how many genotypes we have got? This is one type of genotype. Capital T is one type, small t, and this. So 3 types of genotypes we have got. In what ratio? 1:2:3. Okay. So this is the 1:2:1. Sorry, not 3, 1. Sorry. Clear? So 1:2:1 is the genotypic ratio, what we get in the inheritance of one gene. Clear? So phenotypic and genotypic ratios you have to remember. Now coming to the law, what Mendel has given. This is called law of dominance. First law is called law of dominance. According to this law, characters are controlled by discrete units called factors. Okay, Mendel, whenever he has used, whenever he wanted to describe his laws, he has used the term factors only. He says that something is there in the plants which is getting inherited. Okay, because of this, in F1 generation, only one factor will be, one character will be seen because of that factor only. So like that. So characters are controlled by discrete units called factors. That means what? Character. In our example, it was height. So height is controlled by certain thing which is called factor. This is the meaning. Factor occur in pair. I told you. Homologous condition we have. So these factors, the capital T, are present in pairs. Okay, so factor occur in pairs. This is the meaning. In dissimilar pair of factor. Now, since both of them are similar, we call it as homozygous condition. Clear? And how will be the morphology of this plant? It will be tall because both of them have capital T. But in dissimilar pairs, one factor, if it is dominant, dominates the other recessive. That means in a homologous condition, if one factor is dominant and the other factor is recessive, what will be the morphology here? Tallness. So this is our law of dominance. In dissimilar pair of factors, one factor dominates over the other. Clear? Now, it explains that expression of only one parental character in F1 and expression of both in F2. What do you mean by this? If you look at the experiment, in F1 generation, all the plants were heterozygous condition. That means all of them were only tall. Okay. Whereas in F2 generation, if you look at the morphology, 3:1 we have got, isn't it? 3 were tall and 1 was dwarf. That means what? The dwarfness which we could not see in F1 generation was seen in F2 generation. So both the characters are expressed in F2 generation, whereas only one condition is expressed in F1 generation. Okay. And what is the ratio there? 3:1 is the ratio in F2 generation. So this is all about the law of dominance. Now, for law of dominance, we have few exceptions. What are those? We have 2 exceptions. One is called incomplete dominance, the other is codominance. Incomplete dominance is nothing but our blending. Okay, I have given you the example before. What is that? We have 2 types of flowers. Let us say in snapdragon, which is nothing but antirrhinum. Okay, in snapdragon, we'll see 2 types of flowers, red and white. Now, red flower will be denoted by capital R, capital R. White flowers will be small r, small r. In F1 generation, if you take out the gametes, we have capital R, capital R. These are the gametes we get, isn't it? Small r, like this. Now, if you cross the flowers here, in F1 generation, we get what? Heterozygous condition. All of them will be heterozygous condition only. Okay, and this heterozygous condition, according to law of dominance, it should be what? Red. But in Snapdragon, that is antirrhinum, instead of red flowers, we were getting pink flowers. So pink is formed when you blend red with white. Isn't it? Have you blended the colors in your childhood? Yes. When you mix the red color with white, we get pink. So here we are getting pink color. So that is called blending. Okay. And now when these pink flowers were selfed, when self-crossing was done, again, we have seen the genotypes like this. So the phenotype and the genotype was different. In law of dominance, what was the phenotypic ratio we got? 3:1. 3 were tall and 1 was dwarf, isn't it? But in case of the law of incomplete dominance, we get a different ratio here, 1:2:1, because like red will be your homozygous capital R, white will be homozygous recessive, and heterozygous R and R will be your pink color. Clear? So phenotypic ratio will be 1:2:1. This you have to remember, which is different from that of your law of dominance. Again, coming to genotypic ratio, this is same, isn't it? Genotypic ratio will be same, but phenotypic ratio is different for incomplete dominance. Next, we have something called codominance. Codominance is a condition where both the alleles will be representing their character. If it is present in a homologous condition, if both the alleles are present, okay, two alleles are present like this, both of them will show their character. Example is your AB blood group in humans. In humans, we have different types of blood group, A, B, AB, and O, isn't it? Out of all four, the AB blood group in humans will represent the codominance, because here one chromosome will have A allele, the other will have B allele, and both of them are, dominant on each other. So both of them will be expressed there. Clear? And that is called codominant. That means co is what? Together. Together they will be dominating. Clear? So these are the two exceptions for law of dominance. Next, we'll talk about the multiple alleles. So till now, what we have discussed? We have discussed something called inheritance, then variation, and about the Mendel, isn't it? And why his works were recognized, why his works were unrecognized, failure and everything, why he selected Pisum sativum. And then we have seen the table for what are the dominant characters and the recessive characters, isn't it? And then we have seen the inheritance of one gene. And due to this inheritance of one gene, Mendel could give law of dominance. Now, this law of dominance will have few exceptions. Two exceptions you have to remember. One is incomplete dominance. The other is codominance. Example of incomplete dominance. Now, what do you mean by incomplete dominance? Here, instead of showing up only one character in F1 generation, we have an intermediate character. Okay, neither of the parental character will be seen in the F1 generation. Like red and white flowers, suddenly we'll get a pink flower. That is your incomplete dominance. So we see blending there. Next, we have codominance. That means both the allele together, they will be dominating on each other. Clear? Next, we'll talk about something called multiple allelism. Now, what do you mean by multiple allelism? Here, any character, like if you consider one character, blood groups in humans, the blood groups in humans are actually determined by 3 alleles. Now, what are those 3 alleles? Now, allele will be your I. Okay, it will be IA, then IB, and this is the allele are nothing but genes, as I've told. Okay, and I naught. Clear? So these are the 3 alleles by which we can denote our blood group. So usually, like common person will not call I there. It is simply, they'll say A. So if both the chromosome, let us say this is homologous chromosome. In both the homologous chromosome, if you have A and A, then that person will be A blood. Okay. If in the homologous chromosome, you have B and B, then the person will be B blood. If the person has IO and IO, that is O and O, then the person will be O blood group. And what is the fourth kind of blood group we have seen already? If a person has both A and B, then the person will be AB blood group. Okay, so here a person, if an individual, if you see, if one person, if you see, he can have only two types of alleles. Either he can have A and B or A and A, or a person with blood group A can also have a condition like A and IO. Okay, so person, individual can have only two alleles, but this blood group, okay, the process of blood group in humans can be determined by three alleles. What are those three alleles? A, B, and O. Understood? So these are the three alleles by which the blood groups are determined in humans. Please remember, multiple allelism can be seen in a population. If you have a single human being, a single person, if you have, you cannot see multiple allelism. Yeah, in a single person, we can have only two alleles. But when can you see the three alleles of a character in a population? A person, let us say, in one person, he has A and B. Okay, in another person, he has B and B. Other person has A and A. So if you look at a group of population, in that group of population, we can have all the three alleles. But in a single individual, we can have only two alleles. Okay, this you have to remember. So multiple alleles, example will be human blood group. Okay, each human, that means individual human, will have only two alleles for determination of blood group. But a population will have three alleles. Clear? Next, coming to Mendel's second law. According to Mendel's second law, it is nothing but law of segregation. What do you mean by this law of segregation? I'll explain this here. Let us say we have the homologous chromosomes. Okay. Now, let me write this chromosome number 1. Let me write chromosome number 1. Okay. Now, during gamete formation, please remember the second law and third law, both of them are applied, but they are applicable only during gamete formation. So, you have to start the statement from during gamete formation. Clear? So, the law of segregation is stated as during the gamete formation, the two alleles, okay, the two alleles, or we can say two chromosomes, must segregate independent from each other. What happens here? Out of these two homologous chromosomes, when gametes are being formed, okay, one chromosome will go in one gamete, the other chromosome will go into another gamete. Isn't it? We know that. For example, a person with 46 chromosomes, okay, out of this, 23 chromosomes will go into one gamete, the other 23 will go into the other gamete. So what is happening here? Chromosomes are getting segregated irrespective of each other. Okay, if this chromosome, let me take another example here itself. Okay. So here I have taken chromosome number 1. If I consider one more set of chromosome, let me take chromosome number 2 and chromosome number 3. Okay. So this is chromosome number 3. Now we have a homologous chromosome. Now I'm talking during gamete formation. So how these chromosomes are getting segregated. What happens here? From chromosome number 1, let us say this chromosome is moving this side and this chromosome is moving this side. This we already know, isn't it? From chromosome number 2, this can go this side and this can go this side. Or what happened? I mean, what can happen? This chromosome can move this side and this can go here. So anything can happen. In chromosome number 3, instead of this going here, this will move here and this will move here. All right, so what is happening? In a pair of chromosome, okay, they can segregate independent of each other. It is not like, let me write like paternal and maternal here. Paternal, maternal. Here also paternal and maternal. Paternal, maternal is what? Paternal is from father's side, maternal is from mother's side. So I have taken 3 pairs of homologous chromosome, okay, in an individual, paternal and maternal. So during gamete formation, what is happening? Let us say from first chromosome, paternal is moving to this gamete, gamete A, and maternal is moving to gamete B. Okay. From chromosome number 2, where is 2? This one. Paternal, again, okay, paternal has moved here. Now here also paternal is moving to this gamete and maternal is moving to gamete B. Whereas in chromosome number 3, what is happening? Paternal is moving this side and maternal is moving this side. So there is no hard and fast rule that only paternal will move to one side and only maternal will move to the other gamete. Okay. Any of the chromosomes can move to any of the gametes. So they can segregate independent of each other. Okay, this is all about your law of segregation. Now, I have explained this in terms of chromosome. You have to just replace the term chromosome with alleles or factors when Mendel was using it, because Mendel didn't know about chromosomes, isn't it? So what is law of segregation? During gamete formation, the 2 alleles of a gene, okay, the 2 alleles of a gene. Now, in the chromosome, we have genes, right? Like this. So, this gene or this allele is moving this side and the other gene will move to the other side. So, the 2 alleles of a gene must segregate independently from each other. Okay. And please remember, this is a universal law. That means we don't have any exceptions for Mendel's second law. All right. This is all about the second law. Next, we'll talk about something called test cross. Test cross is used to test the genotype of a plant. For example, I have given you 2 plants. Two plants I have given you. Both of them are tall. Okay, so let us say A and B plants I have given. And I haven't told you the genotype of A and B. Now, I'm asking you to find out, both of them are tall. Let me write down that also. Both of them are tall. Okay, now I'm asking you to determine, to find out which of them have got homozygous and which of them has heterozygous. Homozygous is what? Capital T, capital T. Heterozygous is this. Clear? If both the condition, in both the condition, the morphology will be tallness only. So I've given you 2 plants and I'm asking you to find out the genotype of this tall plant. What will you do? You will do the test cross. So you are basically testing the genotype of the tall plant. Just remember like that. Okay. And how will you do that? Simply, both the plants you have to cross with a dwarf plant. So both of them will be crossed with a dwarf plant. And the result, what you get, will be different. So based on the result, we can say which is homozygous and which is heterozygous condition. So in this, let me consider the first here. If it is homozygous, if you cross it with the dwarf plant, all of them will be tall. So if you are getting progeny, all the tall plants, then the plant is your homozygous tall. Okay. Instead, if you are crossing a plant, tall plant with a dwarf, and you are getting both tall and dwarf in 1:1 ratio, then the genotype of the tall plant will be heterozygous. Are you following my words? Genotype, heterozygous, homozygous. Okay, please don't get confused between all these terms. Or I'll make a separate video to explain the terminologies which are used in this chapter. Okay. All right. So this is about the test cross. Very, very important from your point of view. You have to remember where we use this test cross. Yeah. Test cross is used to determine the genotype of the dominant character. Yeah. Next, we'll talk about the inheritance of two genes. So till now, we have been discussing about the inheritance of one gene, isn't it? Which was giving us the law of dominance and law of segregation also. Next, we'll talk about inheritance of two genes, which will give us the third law, that is independent assortment. So here what have happened? What is happening? We have to consider two characters. So instead of taking only height, I can take one more character. Okay. So in order to make it simple, in seed, okay, in seed we'll be learning two characters. What is that? Shape and color. So seed, shape, and seed, color. So both these two characters, if I consider at a time, then we call it as inheritance of two genes. Clear? Because each gene is responsible for one character there. So inheritance of two genes. When how they are getting transferred and everything, we are seeing the two characters we are taking. Hence, this is inheritance of two genes. Okay. Now coming to your table again, parents. Now here again, I have taken, not I, Mendel has taken pure lines. Just now I explained what is pure line. Nothing but homozygous condition of the parents. When we get, how do we get the pure line? When the parents are repeatedly self-crossed for several generations, the sixth or seventh generation will be completely homozygous condition. Okay. So pure lines we have to take. One will be dominant, the other will be the recessive. Okay. Now here I have taken the shape and color. All right. So what are the dominant shape of the seed? Round. And what will be the dominant color? It is yellow. It is not green. Please remember. So round and yellow seed will be dominant and wrinkle green seeds will be recessive. Clear? Now, coming to gametes. Now, again, I have seen many students getting confused how to take the gametes. Now, here, since both of them are homozygous, it is easy to take the gametes. See, this R will go with Y. So, we have RY. Okay. Similarly, this R will go with this. So, we have RY. Now, again, we'll go with the second R. This R goes with Y like this. And this R goes with Y like this. So we are getting four gametes, but how are those four gametes? All of them are same type. Okay, and that is the reason why I have written only one type of gamete here. Clear? Similarly, here also I am getting small r, small y. So all of them are same. Clear? Now, if you talk about F1 generation, what I have done? I have taken the gametes. Okay, and then I have made the Punnett square. How will be the progeny? According to law of dominance, all of them will be dominant, heterozygous. So heterozygous I have got everything. All the progeny are heterozygous and everything is round and yellow. So F1 generation is heterozygous dominant. 100%. Okay. Next, coming to the selfing again to get the F2 generation. Now here comes the problem. How to get the gametes here? Again, use the same trick here. This is our parents. Okay, these are the parents we have. These are the parents. Clear? Now from this parent, I have to get the gametes. So what I have to do? This R will go with Y. So capital R, capital Y. Again, this R will go with this Y. So capital R, small y. Okay. Then again, small r will go with this Y. Small r, capital Y. And small r will go with this Y. Small r, small y. Okay. If you understand the formation of gametes, trust me, this selfing and this, loss and everything is very, very easy for you to understand. So this is how we get four different types of gametes. If you look at this, we have got four different types of gametes here. All of them are different. So put all those four in one row here. Okay. Similarly, now since both the parents are same, now the other side also you put all the four different types of gametes. and then you make a Punnett square and then you cross it. Okay. So here, what are the things we are getting now? Again, you have to look for the phenotypic and genotypic ratio. Here, phenotypic ratio, we are getting 4 types of phenotypes. Round yellow is what? Both of them are dominant. Okay, round is dominant, yellow color is dominant. Wrinkle green, both of them are recessive. Clear? And what is the ratio there? 9 are round yellow, 1 is recessive. Now, what is this 3 and 3? You have to mix and match there. Round is dominant, green is recessive. Okay, so round, recessive, dominant, recessive, recessive, dominant. Round green, wrinkle yellow. Clear? So phenotypic ratio will be 9 is to 3 is to 3 is to 1. Very, very important. And based on this, many numericals are asked. Numericals, I'll make a separate video of that. Okay. Next, coming to genotypic ratio. Now, genotypic ratio is, something called 1 is to 2 is to 1 is to 2 is to 4 is to all those things. So I'll give you a trick to remember what kind of plants are seen in genotypic ratio. See, this you can easily remember. First, I'll tell you how to remember. First, you remember 1, 4, and 1. Okay. You have to remember that we'll get 4 number, you can remember, no? 4. We'll get 4 1s and 4 2s. Okay. Did you get my point? Let me write with some other color. Everything is same color. Let me use some other different color. See, you'll get 4. You get 4 2s and you get 4 1s. These are the things you get in genotypic ratio. All right. Now, I'll tell you. 4 is your heterozygous condition. What is that? This one. Heterozygous condition will be your 4. Clear? Now, out of 4 1s, 1 will be completely homozygous and the other one will be again. Now, see, one more trick I wanted to tell you. 1s will be homozygous. 1s will be always homozygous. 4 and 2s will be heterozygous. I'm giving you a trick to understand and remember. Clear? 4, both the characters are heterozygous. 1s will be homozygous. So this is the condition where both the characters are homozygous, dominant. This is a condition where both the characters are homozygous, recessive. Clear? Now, coming to now this you can remember. Let me write again. Out of this, okay, 1 is to 2 is to 1 is to 2 is to 4, all those things. 4 will be your heterozygous completely. First one is homozygous dominant. Last one will be, homozygous recessive. Okay, like this. Now, coming to the other ones. I told ones will be always homozygous. Okay. So here what happens is capital R, small y, okay, and small r, capital Y. See, these are also homozygous only, no? Small r, capital Y. Okay. So whenever we have homozygous condition asked the genotype, it will be always 1. Clear? So this one, let us say this is capital R, small y. And this is small r, capital Y. Clear? Now, coming to the 2s now. What happens for 2s? It is heterozygous. Clear? So 1, 2 will be like this. All right. One of the character will be heterozygous. You have to remember like that. Similarly, this is again heterozygous, but small y. Here we have capital Y. Here we have small y. Now you keep the R's same and, go with Y's there. So what happens? Capital R, then Y will be heterozygous here. Again, smaller, Y will be heterozygous. Am I clear? See this, if you practice this for at least 2 to 3 times, you will get it. And there will not be any problem to remember this for the exam. Clear? So please follow this trick. You again rewind the video and then you go with this trick. So this is what we see for the inheritance of the 2 genes. Clear? And because of this, as a result of this inheritance of 2 genes, Mendel has given the third law, which is called law of independent assortment. Now, what do you mean by law of independent assortment? Alleles of genes located on different chromosomes assort independent of each other. Now, what do you mean by this? Let me consider here chromosomes. This is chromosome number 1, okay? And then this is chromosome number 2. I have taken 2 chromosomes, homologous chromosomes I have taken. Now, I am going to write the genes here. Let me write genes. Let us say here I have capital A, both of them are homologous. Then I have B and heterozygous condition. In chromosome number 2, let me take some other pretty color. Okay. So here I have capital C, small c, capital D and small d. Okay. Now if this is the condition of the genes and during the gamete formation, okay, what did I say for law of segregation? I said from chromosome number 1, this chromosome will go this side and this chromosome will go this side, okay. And from chromosome number 2, this can go this side and this can go this side or vice versa. Clear? So in law of segregation, please remember the complete chromosome is moving towards the gametes. But in law of independent assortment, what happens is within the chromosome, now when during meiosis, if you remember, we see something called recombination. Okay, exchange of genetic material between the homologous chromosomes. So as a result, what happens is, let me erase this. So during recombination or after the exchange of genetic material, in this within a homologous chromosome, that is chromosome number 1, okay, this A can go in this gamete and this A, it is going to another gamete, okay. If recombination has not taken place, now capital B will go into this gamete and small b will go to this gamete. But if recombination has taken place, what will be the other condition we can have? Okay, this is one condition of gametes. AB, capital A, small b. Okay. If recombination has taken place, so what happens there? This A can go with this. Okay, exchange of genetic material. B has shifted here. And capital B has moved here. So this A will go with this. And here again, B. Okay, so this is how we can get different gametes. Again, in chromosome number 2 also, okay, the same thing will happen. So let me explain that before that. So here, within a set of homologous chromosome, what is happening here in law of independent assortment? We'll be talking within the chromosome, what is happening to the segregation of the alleles. Please don't get confused between law of segregation and independent assortment. I've seen many students getting confused. Law of segregation, that is second law, is related to your chromosomes. So entire out of these 2, let us say these are the 2 homologous chromosomes, okay? During gamete formation, this complete chromosome will move to one gamete and this complete chromosome will move to another gamete. Clear? But during law of independent assortment, that is third law, what happens? When there is no recombination, that means when there is no exchange of genetic material, whatever genes are present in this chromosome will move to one gamete and this chromosome will move to this gamete. But when there is recombination, let me consider again. let me take this exchange of genetic material. So this cap of this gray pen has moved to this cap. Okay, like this. So let us say exchange of genetic material has taken place. Now the chromosome is different and the gene is different now. Initially, this pink one was present here. I have exchanged this. Okay. So what happened along with this gene? Now a new gene is going towards a gamete. Am I clear? Okay. So within a individual set of homologous chromosome, when the sets, like how the alleles are getting assorted, that is discussed in law of independent assortment. All right. Now here we need not have only two, we can have several genes there. When the recombination, when the exchange of genetic material has not taken place, all the genes will at a time go to one gamete. Instead, on the other hand, if there is recombination taking place, now this gene, which was actually present in this chromosome, has come to this chromosome. As a result, this gene is moving along with the gene which was already present in this chromosome. Clear? Okay. So here, law of independent assortment is something related to the assortment or segregation of alleles of the genes, whereas law of independent assortment is segregation of the entire chromosomes. Please try to remember the difference between these two. And one more difference, law of segregation do not have any exception. It is a universal law because a chromosome as it is moves to the gamete. But for law of independent assortment, that is third law, we do see the exception, and that exception is called linkage. Okay, so as I have told, if the complete genes, whatever we have seen here, if it is going to one chromosome without any exchange, then we say that genes are linked to each other, and the process, we call it as linkage. Okay, I'll explain here. So here, let us say these are the two homologous chromosomes we have. Now on this chromosome, let me consider these are the two set of genes we have. Okay, A and capital B, small. Clear? So now if you look at the distance between these genes, what is there? The distance is very, very less, isn't it? So when the distance is very less between the two genes, if I consider, let us take this chromosome, okay? So within a chromosome, if the distance between the two genes or the two alleles, cannot say two alleles, two genes is less, then such genes are called as linked genes. Okay, why they are linked genes? Because they are very close to each other. In a chromosome, if the two genes are very close to each other, such genes, we are calling them as linked genes. And whenever they are involved or whenever they are forming the gametes, when these chromosomes are getting segregated, okay, as I've told in law of segregation, chromosomes will get segregated, isn't it? Now this chromosome will go here and this will go here. So in this gamete, both small a, small b will come, and in this gamete, capital A and capital B will come, always, because both of them are very, very tightly or closely packed with each other. Clear? So this process of transfer or inheritance of linked genes into the gametes, we call it as linkage. See, this process is very easy, but I don't know why students feel it difficult. First, you have to understand what do you mean by linked genes. What do you mean by linked genes? The genes which are very close to each other are called as linked genes. Okay. In this example, A and B are very close to each other. We cannot see any, exchange of material, exchange of genes during meiosis. They are definitely very close to each other. And when these linked genes are entering into a gamete together, we call that process. So linkage is a process. Yeah. linkage, we can say it is a process of transferring or assorting of linked genes into the gamete. We call it as linkage. So whenever we have linkage, we don't see independent assortment. Now, again, coming to independent assortment. What was independent assortment? In independent assortment, this capital A can go with this, or this capital A can go with this small b. Okay. But when the genes are linked, that means when they are tightly packed to each other, the capital A, can it go with this small b? Yes. During linkage or during linked genes, when the genes are linked with each other, can this capital A go with small b? No, we cannot see that because there is no exchange of genetic material. So capital A will always go with capital B only towards one gamete. Such process is an exception for law of independent assortment. Law of independent assortment says this capital A can go with this or this. But linkage say no, capital A has to go with only capital B, not with small b. Understood? So this is all about the laws of Mendel. Okay. Now we have another topic called pleiotropy. Now what do you mean by pleiotropy? When a pair of alleles control more than one character or phenotype. Simply when a one gene or a single gene controls many characters. Okay, then such concept or such process, we call it as pleiotropy. Clear? When a pair of alleles is what? Nothing but a gene control more than one character or phenotype. Understood? So one gene controls many character is pleiotropy. Examples, phenylketonuria. Phenylketonuria is a type of disorder. When do we see this phenylketonuria? Usually, okay, normally what happens in our body, we have a phenylalanine, which is an amino acid. So this phenylalanine gets converted into tyrosine, which is another type of amino acid. Clear? Both of them are aromatic amino acid, if you remember the biomolecules. So here, conversion of phenylalanine into tyrosine requires certain enzyme, and that enzyme is our phenylalanine hydroxylase. Okay. Now this is an enzyme, and enzymes are made up of proteins. Isn't it? We know that enzymes are made up of proteins. And how do we get proteins? Molecular basis of inheritance. Yes, gene expression. From DNA, we get transcription, and from there, we get the translation process to form proteins. So basically, this protein or enzyme we are getting from DNA. Okay. So in DNA, we have genes. Is it? A molecular basis of inheritance is over for us. You can go through my videos there. So we have discussed everything. Now, the gene responsible for this enzyme, that is phenylalanine hydroxylase, will get defective. That is, it will get mutated. In this DNA, okay, the gene which is responsible for the synthesis of this phenylalanine hydroxylase will get mutated. Mutated is what? It will get defective. As a result, what happens? Can we get this protein? No, we cannot get this protein, or we cannot get this enzyme. So what happens? As a result, phenylalanine is not able to get converted into tyrosine. Isn't it? And this phenylalanine now gets, accumulated in various parts of our body. It can get accumulated in our brain, then liver. Like that, it can accumulate in various parts of the body. And as a result, we see different conditions like mental retardation, reduction in hair, skin pigmentation, all those things. So basically what is happening, if this gene was functional, okay, if this gene was functional, we could get phenylalanine hydroxylase. As a result, phenylalanine is getting converted into tyrosine. Okay, so we are not seeing, we can see a healthy mind, that is healthy brain, then good growth of hair, and we don't have any skin pigmentation like that. But when could we see the actual function of this gene? when that gene is mutated, we could see mental retardation, reduction of hair, and skin pigmentation. So these three characters are actually related to this single gene. Understood? So this is called pleiotropy, one of the example. Next example, apart from phenylketonuria, is taken from the pea plant itself. There is a gene, okay, B gene. So we have capital B, like this. So this B gene is responsible for two characters in pea plant. This will give you the shape of the seed as well as the size of the starch grain. See, grains will have starch in it. We have proteins. Apart from that, we have starch also. So this single gene, that is B gene, will give both shape as well as the starch grain, size of the starch grain in the seed of the pea. Clear? So character 1 is shape, the character 2 is size of starch grain. Clear? And here, depending on the type of genotype also, the things will differ. See, here we have an interesting thing here. What is happening? If the gene or the alleles are homozygous, we see the shape of the seed will follow your law of dominance. Okay. If it is homozygous dominant, it will be round. Homozygous recessive, it will be wrinkled. And if it is heterozygous dominant, it will be again dominant. Heterozygous condition, it will be dominant. That is, round is a dominant, round seed. Understood? But the same gene, if it is present in this genotype, okay, what happens to the size of starch grain? If it is homozygous dominant, that is capital B, capital A, it will be large. If it is homozygous recessive, the starch grain will be small. But if it is heterozygous, instead of large starch grain, we are getting intermediate. So can you tell me what kind of concept we see here? It is not law of dominance. Now, what is that? Exception of law of dominance? Is it codominance or something else? Yes? Which concept we are seeing here? Large, intermediate, and small. Suddenly an intermediate thing is coming out. Blending is seen here. Isn't it? So this is incomplete dominance. Example will be for incomplete dominance. So we can say a single gene, that is gene B, is showing us two types of dominance here. Law of dominance with respect to shape of the seed and law of incomplete dominance with respect to the size of the starch grain. Yeah? So these are the two examples for your pleiotropy. Next, we have polygenic inheritance. Poly means many, genes. Genic is genes. Okay. So what happens here? Many genes will control one character. So we can write many genes control one character. All right. Example will be the human height and human skin color. If I talk about the skin color here, in humans, we have a range of skin color. We can have darkest skin and the fairest, the lightest skin, isn't it? Now, this skin color is actually governed by 3 pairs of alleles. Clear? Now, these 3 pairs of alleles are present in different chromosomes. All right. Now, if all of them are dominant, then we can have the darkest skin color. If all of them are recessive, we can have the lightest skin color. And if you see the combinations of all these things, then we'll have the intermediate ones. Clear? So this is about like the alleles responsible for one character, that is skin color. So here the character is skin color and different genes I told. So what are those genes? A, B, and C. These are the 3 different genes which are responsible for one character in humans. Okay. And one more point they have given in your textbook. Apart from this genes, the skin color in humans is influenced by environment also. Okay, that point you have to remember. Next, we'll talk about chromosomal theory of inheritance given by Sutton and Boveri. Very, very important. Many a times the question is asked. chromosomal theory of inheritance given by Sutton and Boveri. Now, what do they say? See, whatever Mendel has told in terms of factor, Sutton and Boveri will be telling in terms of genes and chromosomes, because factors are nothing but our genes which are located on the chromosomes, isn't it? So whenever they describe the law of dominance or law of segregation or law of independent assortment, instead of using factors, they'll be using what? The genes and the chromosomes. Clear? So they say pairing and separation of pair of chromosomes would lead to pairing and segregation of pair of factors, because factors are nothing but genes. So whenever chromosomes are getting segregated during gamete formation, along with the chromosomes, the factors which are present on the chromosomes will also get segregated. Okay, this is all about the chromosomal theory of inheritance. Next, we'll talk about the linkage given by Morgan. So till now, we were discussing about Mendel's laws, isn't it? Now we'll be discussing about the Morgan. The concept of linkage was given by Morgan. I told you what is meant by linkage, isn't it? Linked genes are what? The genes which are present very closely in a chromosome are called linked genes. Say, for example, capital A, capital B, small b. Okay, so these genes, the distance between them is very, very small. So such genes which are present are called linked genes. Understood? And when these linked genes are moving towards the gametes, okay, as it is without getting segregated, as it is when they are moving towards the gametes, such process we call it as linkage, and this was seen or observed by Morgan. Now, which organism Morgan has used? Drosophila melanogaster, otherwise called as fruit fly. All right. Now, why Drosophila melanogaster? Just like Pisum sativum we have discussed, here also we have to discuss few advantages of using Drosophila melanogaster. This can be grown. Now, this is what? Grown. Okay. This insect can be grown in simple medium. It is usually grown in laboratories. We need not have a complex, fancy items to grow and everything. It can complete its life cycle within 2 weeks. So we need not wait for years together also. And single mating, it will produce a large number of progeny. Okay. And we can have so many individuals again for experimentation. And moreover, male and females are morphologically different. If both male and female are same, okay, in one test tube, say for example, both of them are same, you have put two organisms in this and you are waiting for days together, still progeny is not coming. What will be the reason there? What would be the reason? Can you expect? Yes? Both of them could be females or both of them could be males. If you're not able to differentiate the male and female, simply you're taking two insects and putting that in a test tube and allowing them to mate. Okay, after many days also, we are not seeing any progeny. So what will be the reason there? Both of them would belong to the same gender. Okay. So in order to avoid that confusion, it is better to have morphologically different male and female, isn't it? So that is the biggest advantage I can say here. So male and females are morphologically different. Males will be smaller in size, whereas females will be larger. This point you have to remember. Females will be bigger in size, males will be smaller in size. Okay. Next, T.H. Morgan coined the term linkage. Linkage, we know that. Next, recombination. What do you mean by recombination? Exchange of genetic material between the two homologous chromosomes is called recombination. What is that? Let us say this is a small a, then capital B and small b. Okay. Now this is a condition before meiosis. After meiosis, if we have capital A here, small b here, and capital A here and capital B here. So what happened? This has got here and this has got this side. Here. Now this thing we call it as recombination. Recombination is what? Exchange of genetic material. So these things were observed by Morgan in his experiments. Here. Now what are the results or analysis he has done? Linkage is inversely proportional to the distance between the two genes. Means what? If you have one more gene, let me write C and C here, or capital B. Because both C's will be same, no? So we'll get confused. Okay, so let me consider the third gene, capital D and small d. So what they are telling, linkage is inversely proportional to the distance between the two genes. That means less the distance between the two genes, more will be the linkage. Isn't it? A and D are linked genes compared to, see, A, D and B, all of them are linked. But where do we see more linkage between A and D? Clear? I said A, D and B, all these three are linked genes because all of them are present on the same chromosome. But which of them are more linked? Where do we see more linkage? The ones which have got less distance, that is A and D, will show more linkage. They will go together more often compared to the B. Clear? More the distance, less will be the linkage. Compared to A and B, the distance is more, so there are chances of recombination there. Clear? This is called recombination. So in gametes, we can have capital A, small a, capital B. But whenever we have linkage, strong linkage between the genes, so capital A will always go with capital D and small a will always go with small d. Okay, so between A and D, we can see the linkage, and between A and B, we can see the recombination. I hope you are following. Okay. So recombination is directly proportional to the distance between the genes. More the distance, A and B are farther from each other. So more the distance, more will be the recombination. Clear? Completely linked genes are always inherited together. In my example, completely linked genes will be A and D. Okay, so A and D are very, very close to each other. They will be always inherited to the gametes together. Clear? Now, the experiment done by Morgan in Drosophila, according to them, what are the results? Results are very important here. So genes, now they have taken three characters in Drosophila. Clear? One was the eye color, body color, and wing size. You have to remember eyes, body, and wings. Okay, these are the things they have. Eye color, body color. So colors will be 2 here and size is 1. So these are the three characters they have taken while doing the experiment. Clear? Now you have to know like what is association between these three characters. Very important from NEET point of view. So please concentrate here. Experiment is not important for NEET. This result is important. All right. I've seen many students going behind that experiment and leaving this result. So please don't do that. Eye color, body color, both are colors. Okay. Just remember, both are the colors. They show more linkage. More linkage means less recombination. So here, less recombination, that is 1.3% of recombination is seen. Clear? Why less recombination? Because the eye color and the body color are closely linked with each other. Say this is eye color and this is body color. Both of them are very tightly packed, I can say. Okay, they are very tightly packed and hence there are less chances of recombination. So only 1.3% of recombination you can see them. Whereas eye color and the wing size, the next experiment was done between eye and wing. Okay, so here wing size, okay, so the distance between eye color and wing size was more. And as a result, we could see more recombination. So recombination percentage was 37%. And here the recombination percentage was only 1.3%. Okay, so with this data, we can say that eye color and body color, genes responsible for eye and body color are very close to each other compared to the genes responsible for eye color and the wing size. Okay. So here, coming to the gene frequency again, Alfred Stutwand, who was the student of Morgan, he used the frequency of recombination for gene mapping. Now, what do you mean by gene mapping? Gene mapping is the location of genes on the chromosome. Okay. So based on the distance of the genes in the chromosome, we can tell where exactly the gene is present in the chromosome. For example, he has given the units also, like 1 centimorgan is equal to 1% of recombination. If the amount of recombination, that is exchange of genetic material, is the frequency of exchange of genetic material is 1%, then the distance between the two genes will be 1 centimorgan. It is like, let us take, this is the chromosome here. Okay, now this is gene A and this is gene B. Clear? Now, if the recombination frequency, that means how frequently they are undergoing recombination, exchange of genetic material. That is, here we have small b and here we have capital B. So this is called exchange of genetic material. So if the frequency of exchange of genetic material, that is recombination, is 1%, then based on that 1% recombination frequency, we can say that distance between them is 1 centimorgan. Clear? Similarly, if the recombination frequency is 2%, okay, 2% out of 100 times, if only 2 times they are undergoing exchange, then the distance will be 2 centimorgans. Likewise, if like half of the chromosome, okay, now if the distance is like this, A, and let us consider here we have a gene C. So the recombination frequency, that is exchange of genetic material frequency, is 50%. Now the distance will be say 50 centimorgan. Okay. Now now here I have written one statement that recombination frequency is never more than 50%. So what do you mean by that? Why it is important there? Because see, once you have crossed the half of the chromosome, let me consider the color. Okay, so here we have A and I have one more gene D. Clear? Now I have crossed the 50% of the chromosome structure. Okay. Now, whenever you talk about the exchange of genetic material, if the D getting recombinant, this D coming here and this D going there will be always 50% chances, isn't it? The recombination frequency will be 50%. It will not be 60 or 70 or 80. Clear? So that is the reason why we say that recombination frequency is never more than 50%. Once the uh, alignment of genes or the location of genes is has crossed the 50% of the chromosome, okay, even though it is present at the other tip of the chromosome, let us say E is present here, still the recombination frequency will be 50% only. Okay, so these are the very important points we can say from need point of view with this topic, linkage. Okay, next we have something called sex determination. Sex determination is determination of sex, okay? And here we can have male heterogamy and female heterogamy. Heterogamy means what? Hetero is different. When they produce gametes, okay? When they produce gametes, we'll have different types of gametes. So a condition where an individual is producing two different types of gametes, okay, based on that. So if males are producing two different types of gametes, for example, humans will produce human male will produce sperms and eggs. But human female will produce only eggs. So this we already know. So in our case, males are producing two different types of gametes, eggs and sperms. Isn't it? So we consider them as male heterogamy. So we come under male heterogamy. All right. So here in male heterogamy, we have XX, XY type, XX, and XO type. Here, XX and XY, examples, humans, Drosophila, and some insects. Please remember here we have some insects. Coming to XX and XO. XO, here again, XO will be male. Clear? and XX will be female. So whenever gametes are produced, both the gametes will be XX. Whereas in XO type, what happens is, let us assume grasshopper. Okay, in grasshopper, the diploid number will be 24. So okay, I have written here. So diploid for females in grasshopper is 24. So when this female is producing the gametes, it will produce 12 and 12 because 24 divided by 2. Whereas if you consider the male part, so here males will have 23 chromosomes only total overall diploid. And when it is producing gametes, one gamete will have 12, the other will have 11. So that means one chromosome is less in one of the gamete. Understood? So that is O is one less there. So again, second. Okay, so this was all about your male heterogamy. So humans, Drosophila, and some insects will be showing XX and XY type, whereas the grasshopper, I mean, here you have to remember that many insects will be showing XX and XO type, and example will be your grasshopper. And one more point, males have only one X chromosome. That means one chromosome will be less in males compared to females. Okay. Next, coming to female heterogamy, it is ZZ, ZW, and examples will be birds. So you have to remember all the examples for this. Next type of sex determination is based on the ploidy. It is haplodiploidy condition, which is seen in honeybees. Now, in honeybees, we usually don't count the numbers. It is whether they are diploid or haploid. If the individual is diploid, then that individual will be female. If the individual is haploid, then that will be a male. And males, in case of honeybees, we call it as drones. Clear? Now, if it is a diploid, obviously during meiosis, that is during gamete formation, this diploid female will produce two gametes, isn't it? Whereas males during, we do not see meiosis here because it is only one set of chromosomes. So it will simply undergo mitosis to form the gametes. So for gamete formation, we see only mitosis, whereas for females, we see the meiosis for gamete formation. This point you have to remember. Clear? And as a result of meiosis, we get two gametes, haploid gametes, and from males, that is drones, we get only one set of gametes. Clear? Now what happens? Out of these two sets of gametes, from the females, one set will simply, okay, I have, all right, so let me consider this. Okay, this set of gamete is crossing with the gamete of the male. As a result, we are again getting the diploid, and the remaining set of the gamete, which is haploid, will simply undergo parthenogenesis. This point is very important. It will undergo parthenogenesis to form the males. See here, again, I'll write. This is very clumsy here. See, females are diploid, males are haploid. This is female and this is male. Okay. Females will produce gametes by undergoing meiosis, and males will produce gamete by undergoing simply mitosis. All right, so we don't see reduction division in case of males. Now what happens? Out of two gametes from females, one gamete will go with the males to form diploid again, and the remaining set of the gametes will simply undergo parthenogenesis to form the males. Again, this 2n will be our females, and this single ploidy will be the male. Okay. So this is how we see the sex determination in case of honeybees. That is haplodiploidy type of sex determination. Okay. So this is all about in this video. I'll be continuing this video later. Okay. Thank you.